All right. Hi, and happy Friday, everyone. Thank you so much for joining us today. My name is Casey Volpe, and I manage MBC initiatives at Komen. So first, I just want to say a deep, heartfelt thank you for everyone who registered and is attending live. As I'm watching these numbers trickle up, I'm just so grateful. This is a record-setting event for our program. We have just shy of 600 registrants. And as I'm watching these numbers trickle up, over 100 people on live with us today, which is phenomenal. So whether you are new to the MBC Impact Series and this is your first webinar, or you've attended several, thank you. Thank you for your participation, your feedback. It really does mean the world, and it really makes this program a success. So if you're new, this program is envisioned as a year-long conversation where every month we cover a different topic and you come and select whichever ones are of interest to you. So today our topic is a research update on triple negative metastatic breast cancer with Dr. Justin Balco from Vanderbilt Ingram Cancer Center. And he will speak about his research that he's doing on the benefits and risks of immunotherapy and how his lab is advancing precision medicine through immunotherapy biomarkers. Okay, I see someone can't hear. Maybe if I just talk a little louder. So a couple of housekeeping things just before I get us kicked off. First and foremost, I have to thank our sponsors, because without them, we wouldn't be able to bring you this information every month. So thank you to AstraZeneca, Gilead, GE Healthcare, and Merck as our premier sponsors. And also thank you to Walgreens and Daiichi Sankyo, our signature sponsors. So last housekeeping note, we will welcome your questions during this event. Please put them in the Q&A feature, and we'll be sure to get through as many as we can. Judging by how large the audience is, I'm just going to preemptively apologize for not getting through all of them. I hope we can, but just in case. Towards the end of the program, we're going to put a survey link in the chat. This survey helps us determine topics, cadence. It helps inform the programming that the MBC community finds most valuable. So if you have a few minutes of your time, we really, really are appreciative of any feedback you provide. So now I have the absolute privilege of introducing you to today's moderator, Marlena Murphy. Marlena was diagnosed with stage 3 triple negative breast cancer in 2018, and she maintained a positive attitude throughout her treatment and found herself encouraging friends upon hearing her news. Marlena found her way to the advocacy world because of her desire to help others with cancer. So she began serving as a patient advocate in 2019 with My Style Matters while undergoing treatment. She now currently works as a program manager for GRASP, which is guiding researchers and advocates to scientific partnerships. She works as the community advocate for Turning Point Breast Cancer Rehabilitation and serves on the ASCO Clinical Trials Access and Inclusion Task Force. Whew, I'm getting a little tongue-tied, Marlena. Marlena is also a Komen advocate in science and enjoys learning about research and is open about the realities she has been experiencing because of her cancer. As a result of her diagnosis and a desire to help people, she went back to school to pursue her master's degree. And in 2022, her last year of graduate school, she was diagnosed with metastatic triple negative breast cancer, and she's been in treatment ever since. In May 2023, she finished and she received her master's in clinical mental health counseling with certificates in marriage and family therapy and sex therapy. So currently she provides therapy services to individuals experiencing homelessness, breast cancer patients, and survivors in downtown Atlanta. Marlena Murphy's narrative goes beyond overcoming personal adversity. She is committed to advocacy, education, and making a positive impact on the lives of those affected by breast cancer. And we are so, so grateful she's here with us today. So Miss Marlena, the floor is yours.
Thank you so much, Casey. I am happy to be here. I consider this a privilege and an honor. And this is important to me, just because I want to ensure that I'm updated. I'm updated on the research and that I'm educated and that I stay updated in terms of being able to provide others this information. This is about my life, and not just my life, but this is about the lives of other people experiencing a triple negative metastatic breast cancer diagnosis. And it's in honor of those who I know that have passed away and just those who have passed away in general. Research gives us hope as patients, and it allows us to see and understand that there are people out here, researchers out here, who care enough about triple negative metastatic breast cancer that they want to find viable options for us. And so now I'll introduce our speaker, our presenter, Dr. Justin Balco. And so Dr. Balco is currently an Ingram Associate Professor of Cancer Research and Professor of Medicine and Pathology, Microbiology, and Immunology, and co-leads the VIC Breast Cancer Research Program. He has published over 150 peer-reviewed contributions in the field of molecular oncology and translational oncology research, primarily in the breast cancer field. His laboratory focuses on identifying biomarkers and mechanisms of drug sensitivity or resistance in breast cancer and other tumor types, ways to enhance response rates to immunotherapy by targeting cancer-specific signals of immune suppression, and the biological mechanisms of immune-related adverse events to immunotherapies. His laboratory receives or has received funding from the NIH, NCI, NHLBI, the Department of Defense, the IBC Network Foundation, the Jimmy V Foundation for Cancer Research, the Mary Kay Foundation, Stand Up to Cancer, AACR, Breast Cancer Research Foundation, and Susan G. Komen. So I welcome Dr. Balco. Thank you for being here.
Thank you guys very much. It's an honor to be here. I'm going to share my screen here, and I will trust the chat window if you guys are not able to see my slides. Where are we here? Okay, I'm going to assume everyone can see my slides until I start seeing a bunch of chat bubbles pop up. So it's an honor to be here, everyone. Thank you for having me. I am going to tell you a little bit. Marlene, I saw you jump off of your.
Yes, we see the slides and the we see all the slides and then the slide in the middle.
Oh, so you're not seeing the presentation mode?
Not presentation mode. That's what it is.
Okay. Let me fix that really quick. Thank you for alerting me that. That would have been a problem. How do we look now? I think we're good.
Yes, we are good.
Okay, so for probably the third time that I'll say it, thank you very much for having me here. I'm going to try to tell you a little bit about the research that I've been taking part in, and a little bit of my journey and how I got involved in triple-negative breast cancer research, and particularly as it relates to metastatic breast cancer. Let's see. Yeah. So I think Marlene did an amazing job of my probably overly wordy and detailed biography here, but maybe a little bit more of a personal one. So I started my scientific career at a very early age. I was born in upstate New York. You can tell by the way that I rub my chin, I have a little bit more hair there to stroke on my beard, that I was a pretty inquisitive kid in upstate New York. And I started off thinking, I knew that I wanted to do something in the math and sciences. And I originally started off doing pharmacy training. So I thought that I wanted to be a pharmacist. And I was in SUNY Buffalo in upstate New York. I'm sure we probably have people from all over the country here today, and probably a few of you that maybe even be from the upstate New York or the Buffalo area. This is what it looks like about 2 months out of the year. And then this is usually what it looks like, the other 10 months out of the year. So it's a pretty snowy place. This is actually a picture, I believe, from a blizzard of 2001, I think was the year. And it was absolutely insane. So Nashville, which is where I'm at now, doesn't get nearly so much snow, although it seems to cause an equal amount of problems when it does occur down here. After I finished my pharmacy degree in upstate New York, I decided that I didn't want to be a pharmacist, but I liked sort of the field in general. And I was turned to thinking about a research career. And that was basically because while I was in pharmacy training, I did this one rotation with one particular doctor who was a neuro-oncologist. And, we really got to talking about how to solve a really important clinical problem, and without getting too much into the details. And I was just fascinated by sort of like this problem solving approach. And I think that that's always been something that I've been very attracted to intellectually. And so after I finished my graduate work in pharmaceutical sciences at University of Kentucky, I started a postdoctoral fellowship with this guy, Carlos Artiaga, who's a physician scientist. If you guys attend these and attend Komen events fairly frequently, you may recognize him. Probably not from this picture, though, because this was in like the 80s. Of course, he's got a lot more white hair now, but he's an absolutely fantastic mentor and physician scientist, and has been devoted his entire life to breast cancer research. He's now at University of Texas Southwest in Dallas, Texas. So why breast cancer research for me? I think, as I mentioned, I was at a very young age. I liked to solve problems. And I remember hearing on this like 2020 episode about how researchers were looking at sharks because sharks they thought at the time didn't get cancer, and my mom sort of talking to me about this. And I realized that, how big of a problem cancer was. And I had already had, family members, grandparents that had died of cancer at that age. And so I thought maybe, being a very cocky individual at the age of, 7, 8 years old, decided maybe, even though there are lots of experts in the field, maybe I could do something here. And then I actually forgot about that, right? I got involved in I like to play guitar. I like to do sports like a lot of kids. And I was more interested in, girls and things like that all the way through high school. I completely forgot all of these childhood naive aspirations. I then sort of randomly revisited this as an adult. And I looked at it as, when I was in the research field, right, doing my graduate work, I didn't know at that point that I was going to do cancer research or even breast cancer research. But I saw this as an opportunity. So, one thing I'm going to remark on is there's a lot of funding for breast cancer research. And that's because there's a lot of you guys here. And thankfully, a lot of you guys here, right? And so, breast cancer has been sort of on the run for some time. Now, that was the vision that I was given. I know, of course, the reality is we're not there yet, right? There's a lot more work to do. And this is in part by due to the groundbreaking research of others. And it's fascinating to read, all the way through the last 20 to 40 years of scientific discoveries in this area that have led to things like Herceptin and endocrine therapies and, all of the work that's been done before I, even wrote my first paper in breast cancer research. And so that's pretty amazing. And it's really amazing what those scientists and clinicians, oncologists were able to do with a lot fewer tools than what we have at our disposal here in 2024. Probably still a little bit cocky, but maybe a little bit more, a little wiser now that I'm actually a PI. So understand what are the problems, what are the realistic expectations, and how can we get those things done. So we've gotten a lot of opportunities and help along the way. I've listed some of the funding organizations here. I mean, this is, when I mentioned funding, it wasn't because I cared that much about money, but because these are the things that allow us to do what we do and to make discoveries. And ultimately, as a translational researcher, that's what my goal is to ultimately do something that you guys feel that impacts how you are treated. And in order to do that, it takes a lot of funding and a lot of grassroots funding that leads to bigger funding, like things from the NIH or the Department of Defense. So I have gotten a lot of opportunities and help along the way. So one of those things was a Susan G. Komen postdoctoral fellowship, which I got in 2011. I was also awarded a Susan G. Komen Career Catalyst Award, which is this really amazing mechanism that Komen still has today. That is really focused on trying to take people out of these experiential positions, that's a postdoctoral fellowship, and into their own, right, starting their own lab. And then I got some other, as you can see, these things grow into these, national awards like the NIH NCI, the National Cancer Institute and Department of Defense Awards. And these were what really helped establish my lab in 2015, 2016, that really facilitated, a lot of the work that we do today. So this allowed me to form, I put an asterisk next to what we call an independent research program. It's not independent. I've always had an amazing team of people around me. And if I didn't have those people, none of the stuff, none of the things you'd hear about today would have actually, would get done. And so this was the very first team that I formed. These are the people here. They're all Almost everybody here has moved on, except obviously myself and my lab manager, Susan Opalenik, who's really been an amazing person to have around. And I definitely would not have a lab without her. So there's 5 people here, and then I've taken some other group shots of my group. You can see that we're, I wouldn't necessarily call us exponential growth, but we've formed into a much bigger program. Here's the last lab picture that we took. My beard is even perhaps a little bit grayer here and probably a few more pounds extra around the middle here in 2023. But we've grown into a very happy and amazing group of young researchers, many of which will stay in the breast cancer field. So you can, maybe years from now, look on and to another one of these seminars, hopefully given by one of my trainees. So I'm now going to get into some science, right? Because that's the point. It's not about me to talk about, where I came from, but to give you guys a little bit of an idea of some of the research that we're doing in triple negative breast cancer, specifically as it applies to metastatic triple negative breast cancer. So I'm going to start off making sure that everybody has a good, healthy understanding of what T cells are, because my lab focuses on tumor immunology. As you heard in my bio, I'm a professor of medicine, but also of pathology, microbiology, and immunology. So I think after, the COVID pandemic, people are probably pretty familiar, more familiar than they were with some of the different immune cells and immunology of the human body. And so I'm going to talk a little bit about T cells because they're going to be really important and come into play in almost everything that we're going to talk about today. So cytotoxic are one type of T cell, and as the name would sort of imply, if you break it apart, cyto means cell and toxic means poisonous, right? They're a part of our immune system that are defined by a certain protein. We call that CD8. It's not that important. You don't have to remember, but I might mention it again. And so these are really amazing cells, and each one in our body, none of our T cells are the same. So, mine are completely different from every other person, even people in our family. So people, identical twins have less than 1% of their T cells that are identical between the two. So that's how diverse they are. They're not even the same in identical twins. And there's millions of individuals, like little snowflakes, floating through our body, and they scour our body looking for virally infected and or cancerous cells. So they're looking for something that's not self, we say, not self, meaning it is something, but it's not ourselves. These are the types of cells that actually sometimes they get messed up, and they're looking for non-self, but they accidentally recognize self, and that's autoimmunity. So it's one of the elements of autoimmune diseases. This is a really cool little picture just showing a T cell kind of scours the body, it tries to find hopefully a tumor cell here, and then it attaches to it. And I'm going to show you a cool little video. So when these T cells find cells like a tumor cell or a virally infected cell, they can kill them by, you can see this T cell in green. It found this cell and it's sort of feeling it out, almost like, a blind person kind of feeling out the face of someone to get a vision of, is this a normal cell or is this a cancerous or virally infected cell? And once it realizes that this isn't self, it actually opens up little pores and it injects a poison into that cell. And then it moves on to try to find the next cancer cell. So I think that's pretty cool that we can actually visualize some of this stuff. Pretty neat. So one of the jobs in tumor immunology, and specifically tumor immunotherapy, is how we make these cells try to do their job better. And so, this is just sort of a cartoon demonstration of that same idea that I kind of showed you in the last slide, which is, we have these antigens, right? Tumors, all of our cells make lots of little pieces of themselves, and they show them to the T cells that are floating out through the body. And occasionally, those get picked up by certain cells in our body, and they take them to the T cells and they say, hey, look, I found something suspicious. And this causes all the T cells to leave lymph nodes or your spleen and travel through the body and go and try to find that site of inflammation and check it out, make sure it's okay. That could be, a COVID infection or a flu infection, or it could be a breast tumor. And so once those T cells get there, if they realize that this is a tumor, then they might start attacking those cells. But eventually the tumor finds a way around recognition by those T cells. And one of the ways that it does that is by upregulating or increasing the expression of these things called checkpoints. And so these checkpoints are things that are normal cells in our body. Remember that I talked about autoimmunity and that sometimes these T cells get activated against our cells. And we have these secondary mechanisms to try to prevent that. That means that our normal cells will express these checkpoints to tell the T cell to stop and to quiet down. Of course, cancer being cancer will try to hijack these mechanisms. And so one of the really novel discoveries and one of the newest therapies to be approved in triple negative breast cancer are inhibitors of these checkpoints, meaning you sort of restore those T cells' ability to recognize the cancer cells. So just to put this in sort of a little bit more of a translational perspective, both a public perspective and a scientific perspective, you guys are probably all familiar with former President Jimmy Carter, who was diagnosed with metastatic melanoma to the brain. We work a little bit in melanoma. I don't know, how familiar everyone is with metastatic melanoma, but 20 years ago, this was an absolute death sentence. 8 to 9 years later, Jimmy Carter was still alive with a brain tumor. These were amazing drugs that were first approved in breast cancer. These checkpoint inhibitors. I'm sorry, that were first approved in melanoma that are now approved in breast cancer. Not long after that, so after this very public story of Jimmy Carter, Jim Allison and Tetsuko Hanjo from Japan were awarded the Nobel Prize in Medicine. So it used to be many people were awarded the Nobel Prize many years after their discovery, sometimes even posthumously. But this was a pretty quick turnaround for a Nobel Prize. That's how meaningful it was. And I don't want to, it's important to remember that some of these things exist because of money, but just to put a number on it, right? To put a number of patients or put a number of dollars. These are a huge part of our therapeutic field right now. There's indications across all of these different types of cancer. And this number is an easily accessible thing to look at how fast it is predicted to grow, meaning, for each one of these dollars, there's an equivalent number of patients, the cost of that therapy. It's going to increase even more by probably close to fivefold by 2031. So I think I showed you a little bit about these drugs' work. They're monoclonal antibodies that are not unlike, many of you might be familiar with Herceptin, which is one of the first monoclonal antibodies that was approved in cancer therapy. And they block these different interactions between those CD8 positive cytotoxic T cells and either an antigen presenting cell or a tumor cell. Now, something I am going to briefly speak to, if we have time at the end here, is one of the other things that we do that's not necessarily directly related to metastatic triple negative breast cancer, but it is very important for metastatic triple negative breast cancer patients. And that's the toxicities from these drugs. Unfortunately, no matter how hard we try, every new therapy comes with toxicity. Remember, I talked about autoimmunity being sort of the flip side of this process. And as you might expect, these drugs don't have the typical types of chemotherapy toxicity that we're used to, but instead have a very confusing and difficult to understand or predict toxicity. Maybe not. They're autoimmune-like syndromes that can occur really in any organ in a very seemingly random fashion in patients. And so a big part of my lab is to try to understand those things. So that gets me to my lab and what we try to do sort of as our overall mission. So we try to make immunotherapy work better, mainly in breast cancer patients, or mainly with a focus on breast cancer patients. So we try to do that by identifying biomarkers that are able to predict response. And we do that by trying to identify mechanisms of resistance to these drugs and how we can circumvent that by a combination of therapies. And then we try to understand these, what we call immune-related adverse events, which are the toxicities of immunotherapy. So we try to make these therapies safer for patients. So why do we try to understand breast cancer immunotherapy or work on breast cancer immunotherapy? And that's because over the last 10 to 20 years, there's been a number of new what we call molecularly targeted therapies that have been approved. These are things like PI3 kinase inhibitors, CDK4/6 inhibitors, mostly of which are relevant to metastatic ER positive breast cancer. But as a general theme, these drugs are costly. They work only in a population of patients, and ultimately, almost all patients develop some kind of molecular resistance to those drugs. Of course, that's not to be unexpected, or also with immunotherapy. But the one advantage of immunotherapies is that when they do work, they have the possibility of producing very long, durable remissions. You remember I talked about Jimmy Carter, former President Jimmy Carter, and how his expectation of being alive 5 to 10 years later after a brain metastasis with melanoma would not have occurred. There was like a 0% chance that that would have occurred 20 years ago. So you can see what the power of these therapies can do. Now, we knew that breast cancer would not respond, or was very unlikely to respond to these therapies, as well as melanoma, due to historical reasons that I won't get into. But we hope that they would, either with the right combination and the right population of patients, have that capability of producing some very durable responses. And so this is just a graph. It was very early on, one of the very early phase 1 trials, so early in heavily pretreated metastatic triple negative breast cancer, where we started to see that there were some patients that had what appeared to be very durable responses, meaning that their responses would last a long time. And that was really good. And in fact, we know now many years later that there are some patients who can get complete responses. This is a fraction of patients, but can get a really strong response that seems to last years or longer. So how can we help in breast cancer? So I'm going to present really quickly a brief overview of sort of those first two points, biomarkers and mechanism of resistance. So I think this is sort of a two birds, one stone. Not that I want to kill any birds, but this is a quick way for me to sort of describe one of the ways that we addressed both of these points. So it started off with two important questions, and thinking about which patients will benefit to these immunotherapies and how can we identify them using precision medicine. There's a cost of success here. There's also a cost of failure. And secondarily, how do we make more patients respond? So how do we increase that small percentage by combining therapies that might work better together? So this started off with an observation. You remember I told you that T cells love to go through the body and try to identify a virally infected or a tumor cell. So how do they do that? That's a pretty, that seems like a pretty hard task for a cell in your body to, come along and suddenly decide that this cell is not a normal mammary epithelial cell or breast epithelial cell, but instead is a breast cancer cell. And so the way that they do this is by a molecule called MHC. So major histocompatibility complex 1. That's a mouthful, I know. These are synonymous in the human body with something called HLAs. And so just to sort of bring it back to maybe things that people understand, how when we do organ transplants, they want siblings, right? For is probably the best match, but if they can't find that, they will take them from other people, but they do something called HLA typing, meaning they look for how similar your HLAs are, your MHCs are. And of course, siblings are more likely to have very similar. You can kind of think of it as blood typing in a way, but it's actually much more complex. Because there's up to 6 of these that each individual person might have. And there's probably, 40 or 50 different possibilities for each one. So it's very rare to ever encounter 2 people that have all of those 6 MHCs identical. So regardless of that complexity, these molecules are the things that are expressed on all the cells in our body, and they're always showing all of the things that are made in a cell. So if a virus infects a cell and is trying to make more copies of itself, little pieces of that virus are getting chewed up and they're getting put onto these MHC molecules on the surface of the cell. And that's what the T cell comes and identifies and says, okay, is this okay? Or is this potentially a virus or a tumor? Because tumors make other things than what our normal cells do. So we made this observation in looking at this very small clinical trial we had done with immunotherapy here at Vanderbilt. And there were 2 patients on this trial, about 10 patients, that had a pretty good response. They had a partial response. They were metastatic triple negative breast cancer patients. And of these 2 patients, we were staining where this is tissue of the breast samples of the metastatic biopsies. There were 2 patients that had a pretty good response and their tumor shrunk. And when we were looking at all these different markers on the tumors, we looked at this one and we saw that they were 2 outliers as far as most of the tumor cells had lost this MHC molecule. And these 2 had retained it. And that was really interesting to us. Because if you can think, if you're a tumor cell and you're trying to outsmart the immune system, right, and you want to avoid these T cells, the easiest thing to do is to turn off this MHC expression, to silence it. So now you're invisible to the immune system. So we know that these MHC molecules are required to signal to T cells and thus are required for immunotherapies to respond. And I just kind of told you about this process. So I try to make a little, janky cartoon here that kind of shows you that here's the questionable cell, right? And the T cells check self, non-self or self. And so this is this little peptide. Maybe it's a mutation in the cell. Maybe it's not. Maybe it's just a normal part. Maybe it's a piece of a virus. And so this T cell checks and it says non-self or self. But if the cell doesn't have this MHC1, the T cell will never react to it. It doesn't. So that doesn't seem like a very good way to have our immune system. It's an Achilles heel. So there's this other cell type that I'm going to tell you about now. It's called NK cells, or natural killer cells. And as the name would imply, natural killer cells have the ability to naturally kill things. And one of their systems of checks is they check for missing self, right? So this is actually an error on my slide, and I apologize for that. It says non-self. It should say no self or missing self. So the NK cells, they don't care what's on here, right? This little black dot. They don't care whether tumor or not tumor, but if they find a cell that has tried to stop its ability to show things to T cells, then that's where the NK cell will come and kill that cell. So this is a check and balance in our immune system, which I think is pretty incredible. So this got us thinking that maybe these tumors that don't respond to immunotherapy, I showed you here, all of these tumors don't respond because they've lost MHC1. And maybe we can get the natural killer cells to help out in this particular case. So this is just an observation that we made from some more stains. And so, I'm going to show you these, and I know you guys are so used to looking at things like this, I never told you what they are. So these are all cells, right? This is under a microscope, and each one of these colors, pretty neat, is fluorescent signal. So we're able to look at certain markers on the tumor cells, just like a pathologist would. This first one, DAPI, just shows us what's a cell. So these little round blobs of the nuclei of all the cells. This thing called pan-CK or cytokeratin is something that would be expressed on the tumor cells. So you can see all these green things. You can see all the cytoplasm, which is not the nucleus. Everything but the nucleus of the cell is all green. And if you overlaid those, that blue will fill in. It's hard to see here because it's been turned down, but fill in these like gaps. These are the nuclei, these black holes here. And then we have MHC1 or HLA-ABC. It's synonymous, same. And you can see that this one region here has lots of class 1, but this little pocket here has lost class 1, lost MHC1. And that's really big. So that's a big deal for us. We were interested in this, and we said, maybe this is one of the reasons why these immunotherapies don't work very well in a lot of metastatic triple negative breast cancer patients. So we said, let's prove that to ourselves. So we went into the lab, and we made a model. We modeled this in mice with mammary tumors. And this is one of the things that we do. We don't love to do this sort of thing, but I'd much rather learn from the mice so we can confirm it in patients. And so what we wanted to do is say, does this actually cause resistance to these drugs? So we took some cells that were breast cancer cells, and we took those same cells and we used gene editing to get rid of this class 1. So now we have 2 different things that are basically the same cell. And the only way they're different is they have class 1 or they don't have class 1, MHC1. And so we mix them together then. And we asked, what happens if we just even lose 5 or 10% of our cells having no class 1? And it turns out that when it's normal cells, normal cancer cells, they respond really, really well. They don't grow really at all whenever we treat them with this drug that's available now and approved in metastatic triple negative breast cancer. But when we mix them just down to a 5 or 10% spike in of those cells that have lost class 1, now you can see that those tumors start to grow under therapy, meaning that this is causing resistance. So we asked, okay, so given what I told you about natural killer cells, and there were some therapies that are actually in early, clinical development from drugs like AstraZeneca that target these natural killer cells and some of the similar checkpoints on the natural killer cells, one called NKG2A. So we said, okay, now that we know that those tumors are developing resistance, what happens if we leverage that ability of activating the NK cells, right? So now we're using a combination of the cell that targets, the drug that targets the T cells, the one that targets the NK cells, and then now using a combination of both of them. And I don't know if you guys know how to read a survival curve. Basically what this is it's showing how many of our mice are actually are alive and still doing well at each given time point. And so you can see that they all do not very well when they have these mixed tumors. But when we target them with both the NK cell and the T cell targeting therapy, we get mice now that are essentially cured of their disease. And so when we looked biologically at what was going on here, and this sort of illustrates our idea, if you have this mixed tumor where some of the tumor cells have lost this marker and you deliver just this approved anti-PD-L1 therapy, it only kills the positive cells, not the negative cells. Likewise, what we saw in our models was that when we used the NK targeting therapy, it killed all the negative cells, but it left the positive cells. So when we use the combination of both of these, we're able to clean out both of those populations of cells. So we think that this is something that we really want to translate to the clinic. But before we do, we want to make sure we're just working in mice here, right? So this isn't proof of really much other than, we think that this occurs in an alive animal system. So we turn to some investigators here, and I want to point out that this was the work of two really talented graduate students, one Brandy Taylor and Zhao Peng Sun is an international student from China, both working together over the last 4 or 5 years on this particular project. One of the last things we were able to do on this was we wanted to ask in actual patients, metastatic triple negative breast cancer patients, are they going to, do we see some of these same effects? So we looked at a clinical trial that we had done here at Vanderbilt and across a few other institutions in the US where patients had just chemotherapy or chemotherapy plus immunotherapy, that PD-L1 drug that I told you about. And it turns out that patients that were just given carboplatin, which is chemotherapy, if those patients had this mixture phenotype, this mixture of class 1 positive and negative cells, they did equally, I would say equally, not that well on these because it's metastatic triple negative breast cancer. But they did, there was no effect here, the therapy on either population. But when we looked at the patients that were given the immunotherapy, now we saw that those patients that had lots of mixtures of class 1 positive and negative cells in their tumors didn't do that well on the immunotherapy. But the patients that had lots and retained lots of class 1 expression, those patients actually did really, really well. And they the patients who were able to follow, some of them were progression free after several years. So we started to ask, and this is really the next step, and we're looking at trying to do clinical trials now in this area. Can we repeat what we did in that mouse experiment and say, with these patients that didn't do very well and didn't benefit from the T cell targeting immunotherapy, would they now benefit if we combine therapies, right? Those NK cell targeted therapies with T cell targeted therapies. So now I'm going to switch gears. I think we have just a few minutes left here, so I'm going to kind of try to speed through this. It's a little bit less relevant, but I think still really important because everyone knows about chemotherapy toxicity, particularly in a metastatic triple negative breast cancer audience. As I mentioned, these drugs are sort of giving us fits because they have a very different, they're much less predictable. Those toxicities are really bad from chemotherapy. These ones are very different, but the problem is we can't really predict when and how they're going to occur, how they're going to manifest. And so I'm just going to give you one of the ways that we've sort of been trying to work on this. So there's this one really, I think I explained the reason why we see these toxicities with these drugs, these side effects. I'm going to start to talk a little bit and tell you a really quick story here about myocarditis. So carditis, right? Cardio, we would think this is actually inflammation of the heart. And really, this is a very dangerous side effect when it occurs. Thankfully, it doesn't occur very often. It's less than 1% of patients, probably less than 0.1% in breast cancer populations. But just an example of one of the ways we're trying to help in this area. When it does occur, it had nearly a 40 to 50% fatality rate. So this was a medical emergency when this would occur, but again, very rare. So you can see that this is a problem for the deployment of these therapies, particularly when you want to use them earlier and earlier in potentially curable patients. So I think I already showed you this. Any organ system can be affected with these drugs. So, this was a really quick story. This was a really goofy sort of PR picture that we took after. It starts like a bad joke, right? So a cardiologist and a melanoma oncologist walk into a breast cancer research office one day. And these were good friends of mine. And they came in and they said, we had these two patients that, with metastatic melanoma that were treated with these drugs within like a week of one another, both ended up in the ER and unfortunately passed away. And they said, this is a really big deal. We need to figure out what's going on here. And so with some other collaborators, this was something I never thought I would get involved in. Number one, side effects of these drugs. But, they kind of drug me into this. And that paper was published in a journal called the New England Journal of Medicine, which is not something, not a physician, even physicians aspire to, not a physician, whatever, dream of getting a paper in the New England Journal of Medicine. It's one of the oldest medical journals in the world. So I knew that that was a big deal. And it turns out that a couple years later, that guy that I showed you, Jim Allison, who won the Nobel Prize in 2018, came to Vanderbilt to give a talk. And we all met as a group. And he said, I have these mice, and they're similar to those checkpoint inhibitor mice, or those same things are modulated in these mice. And it looks like they're dying of myocarditis. And so we said, we want to work on this. And so this paper went to another big journal called Cancer Discovery, and you can see now we don't have lab coats on because we're a little older and we have a little bit less hair, and they tried to take the same goofy setup for a PR pick. But I had a really talented MD PhD student in my lab, so is now an MD and a PhD, and she's in Washington University in St. Louis, and she wanted to ask, I want to use these mice and I want to figure out what's going on in people. Why are people dying of myocarditis? Again, rarely, but why are these drugs causing this? And so she went all the way through everything that was going on in the mice, and she figured out this really complicated system and went all the way down to that one little antigen, that little black circle that I showed you in that cartoon, of what that was and why some patients were getting this. And so now I think we actually have a way to start thinking about figuring out who is at risk for these really dangerous toxicities to immunotherapy. So with that, I want to bring it to a close. All this stuff, it's really important that we, continue to get research funded, not just my lab, all the breast cancer labs out there, and really any type of cancer. This is what turns us on, what keeps us going, what makes us, do the things that we do and facilitates that research. But most of all, it's these people around in my lab because, I think of some ideas, but these hands don't work very well in the lab anymore. And it's all these really talented next generation of scientists that I get to work with that are able to take, dreams that I have and try to do that, advance those for patient benefit. And the last thing that I just want to remind you is that Balco is OK Lab spelled backwards. So, we're not great, but we're okay.
Well, thank you. Oh, go ahead. I'm sorry.
No, I remembered that I had a question slide, so I can either leave it up or I can close it down for questions.
Well, thank you, Dr. Balto, so much for this presentation and for educating us. We do have some questions. We're going to start with this question. To your knowledge, how much research is going into triple negative apocrine sublevel? Any updates on this sublevel in triple negative breast cancer?
Yeah, so apocrine is a particular type of morphology, we would say. So this is something the pathologists would call apocrine breast cancer. Right now, that we don't have any information that that particular type of breast cancer behaves differently from a lot of different featured breast cancers. So there's some ones that are, are known to be a little bit more bad actors or bad apples, like something called metaplastic breast cancer, which is often triple negative. To my knowledge, Apocrine, really follows the same sort of response patterns and things that a lot of the other subtypes do that aren't treated really uniquely. And that's, not necessarily that part of that could be that we don't know enough about it. But, when we've looked at sort of how these cancers form, it's sort of a nomenclature for them, but it doesn't necessarily mean that they ask to be treated in some very unique or advanced way, at least as of right now. So there's a lot out there actually of ongoing research to try to understand, what some of these different, again, I'll use the term, it's a morphologic features, like what a pathologist would call it, and whether they do need to be treated differently. So far, Apocrine, to our knowledge, doesn't require any specific bells and whistles.
Thank you. Another question is, in your opinion, what's the most significant treatment available now that wasn't available even 5 years ago for those living with triple negative metastatic breast cancer?
Well, immunotherapy. No, I'm just kidding. Actually, 3 or 4 years ago, I would say it was immunotherapy. Right now, actually, one of the most exciting things they're called antibody drug conjugates. So, I was at ASCO, which is a big clinical oncology meeting in Chicago just this past year. I'll go again in a few months. It's in May or June in Chicago, and, got to sit in a crowd where it's only happened a couple times, but got to sit in a crowd and see, big phase 3 clinical trial data release where everybody, stood up and did a standing ovation. So huge numbers of patients that are, looking to benefit from these new antibody drug conjugates that, they're still toxic, right? They're toxic. What they are is an antibody, like you can think of Herceptin, right? Which I think a lot of people in the breast cancer, area are familiar with, by nomenclature, it's one of these antibodies. And it's kind of like combining the chemotherapy with an antibody. So you're trying to deliver that chemotherapy right to the tumor instead. So it still has some toxicity, right? But not quite as much, and much better efficacy than just chemotherapy alone. So I think that's a really, really exciting area.
Another question is, which tests are recommended to see if immunotherapy is or will work? Someone is on chemo and immunotherapy. They had lumpectomy and radiation and will continue on immunotherapy.
Yeah, so this is really under debate right now. So I assume from that brief description is that this was somebody that was diagnosed early stage and got what we call the KEYNOTE-522 regimen, which, kudos to that person for going through that regimen. It's, it's an advance, right, from an outcomes perspective, but it can be pretty rough. And that includes 6 months of immunotherapy after surgery to complete 1 year of therapy. So, there are some studies that are coming out that are being done through large consortiums to try to figure out if that additional 6 months of therapy is needed and in what patients. But right now, we don't know. We don't know. This is an active area of research that we're taking part in. Clinicians and scientists and biomarker developers are all trying to work together, to figure out how, not necessarily to always give less therapy, but to figure out who is really benefiting from that additional immunotherapy. So sorry, I don't have a better answer for that, but it is a very active area of research.
Thank you for the answer. Any trials in the pipeline yet? Do you know how soon they'll be? If so, time is not on our side necessarily with those experiencing or living with metastatic triple negative breast cancer. So that's critical.
Yes. For the idea that I presented, we are in talks, but we don't have a clinical trial scheduled yet. But absolutely, this is our next step for that particular combination of therapies. That said, I would really encourage patients to always be on a clinical trial in that setting, right? Whatever clinical trial, any clinical trial that is an interventional trial is going to give patients at least the standard of care with the possibility of something better. And so, you can't lose in that particular situation. So I really encourage to get to an academic medical center near you, and to find the right clinical trial. Obviously not knowing anything, I can't recommend, any particular clinical trial, but, I would put a plug for some of the antibody drug conjugates, which are just now getting approved in what they call HER2 low breast cancer. But there's a number of these coming down the pipe, and they are in different indications and settings and different lines of therapy. And they're giving good responses.
Another question is, what type of chemos work best with immunotherapy for metastatic triple negative?
Yeah, so we don't know. Interestingly, the one that, the immunotherapy pembrolizumab that's approved in that setting, the clinical trial that got it approved had like 3 different chemo regimens, and they were all equally efficacious, right? So one of them was carboplatin and gemcitabine. One of them was, I think, paclitaxel, and the other one was a drug called nab-paclitaxel, so very similar. And so they all benefited regardless. There are some ideas that drugs like doxorubicin are sort of theoretically better, but there's no real convincing data. Yeah, I will say that there's a few clinical trials out there that are just starting up that combine immunotherapy with those antibody drug conjugates, and that promises to be a really powerful combination.
What are you most excited about in terms of the research, the current research that's underway for triple negative metastatic breast cancer?
Yeah, so I think the areas that I'm the most excited about is, right now, I can't think of the right word. I'm cautious because we have approvals. We have very broad approvals for immunotherapy in breast cancer, but we have some, what we think are really compelling biomarkers that are being tested in large clinical trials now that I think is really going to help identify the patients that really can benefit from that immunotherapy. And that is really going to make these an effective therapy versus ones that, you start to have to balance all these toxicities against. Only about 8 to 10% of triple negative breast cancers actually respond to this drug. So, but if you could identify a group that's 90%, right, or 80%, now that's a really exciting group of patients to treat with these drugs. So that the second area, again, and I hate to be a broken record, but that second area is all of the new things that are coming down with these antibody drug conjugates and tying new therapeutics to them, because they all seem to be working pretty well. And so I think that this is going to sort of open up a whole new toolbox for treating metastatic triple negative breast cancer. And the last thing is, some newer therapies, some new immunotherapies, things like, you may have heard of them, called CAR T cells, right? These are where you take the T cells out that we talked about out of the patient's body, and you modify them to make them really aggressively go out and find that tumor cell and attack it. And I think that these are, they work, they're approved for hematologic malignant cancers. But we've never been able to get them to work in solid tumors. And I think that there's some really cool new advances that are coming out in CAR T cells that are going to allow these therapies to work in breast cancer patients as well.
So along the lines of that, specifically, once Keytruda has stopped working, are there other immunotherapy options that are approved right now for triple negative breast cancer, to your knowledge?
Fortunately, the only one, right? Now we're getting near that leading edge, the cusp of state of the art. And we are waiting for, trials that advance on these. We have one going on through a consortium called the TBCRC, the Translational Breast Cancer Research Consortium. That's about 20 institutions across the country that's looking at combinations of that doxorubicin that I mentioned with immunotherapy to reverse resistance, and a couple other drugs, including one of the antibody drug conjugates called sacituzumab. And so, again, this isn't a new immunotherapy, right, that's approved, but one of the ways that we can use the things that we have approved now is to try to find how we can combine them to reverse that resistance once it develops.
Well, thank you again so much. I am going to hand this back over to Casey.
All right, I'm going to stop screen sharing here.
Perfect. So first and foremost, thank you to Dr. Balco and to Marlena for just a phenomenal presentation and discussion. We had a very active audience, which I absolutely love to see. I love watching you guys share your stories and connect in the chat. The downside is, of course, we're getting questions from every angle and it's very hard to keep up with. So we did our best. I know we got through something like 15 or so questions. If we did not make it to yours, I truly, truly apologize. We weren't ignoring anyone intentionally. It was just the interest was too great. And Dr. Balco had too much interesting things to say. So thank you for a phenomenal presentation. Today's session was recorded and it will be posted and shared out. If you registered, you'll receive it via email. So please go to Komen.org/MBC series and you can check out the entire series. So pick the topics that are interesting to you or relevant to your personal journey and share with anyone that you would like to learn more with. Once again, I'm going to acknowledge our 2024 Impact Series sponsors. So thank you to GE Healthcare, AstraZeneca, Merck, and Gilead, and then Walgreens and Daiichi Sankyo as well. I did drop that survey link in the chat. Please just take a couple minutes and give us your feedbacks to help us continue to grow and do better. And more importantly, make sure we're providing really relevant and valuable information to all you guys. So the next MBC Impact Series event will be March 20th and will be a discussion on dealing with MBC day to day. And it will be 3 women who are currently living with the disease to talk about what they've learned, their takeaways, and answering your questions as well. They've been living with metastatic disease for a variety of time. So they've got all little different perspectives on it. So bring your questions and we look forward to having you log in and join us. So thank you again to everyone. This was incredible. Have a lovely weekend.